Receptors are an important part of cell-to-cell communication. Without receptors, none of our bodily systems would work properly. There are receptors located in the plasma membrane as well as in the cytoplasm of the cell. Hydrophobic signal molecules bind to the receptors in the cytoplasm, and hydrophilic signal molecules bind to the receptors in the plasma membrane and undergo signal transduction. I'm a receptor.
I'm a ligand. Ready? Why are receptors important?
Amanda, they are a key component in cell-to-cell communication. Without the receptors, the ligand will not be able to bind, resulting in no response. This is vital in multicellular organisms to allow the cells to communicate and work together as a tissue, organ, and as a whole organism. Receptors are highly specific proteins with high affinities or attraction to the ligands meant to bind with it. There may be receptors for the same ligands on different organ or body structures that yield a different response. The difference in response is likely due to the signal transduction that often involves a kinase cascade different in each type of cell. A type of receptor for ligands that are hydrophilic are Toll-Like receptors. Toll-Like receptors are transmembrane proteins found on the plasma membrane of macrophages, mast cells, and dendritic cells. For example, this picture describes the affinity a ligand will have for its receptor. Binding affinity is the amount of attraction a ligand has for a receptor. In this picture, the ligand to the left fits perfectly with the receptor and results with a strong bond between each other and a strong cellular response. The ligand to the right shows a weak bond with the same receptor. This will result in the ligand not staying attached to the receptor and yielding a weak cellular response or none at all. These are the three most important cells that are involved in the innate immune response. They act as the first line of defense. I'm a Toll-Like receptor.
I'm a pathogen.
The receptor is 700 to 1100 amino acids long. Toll-Like receptor ligands are from the pathogenic host. It can consist of LPS found on gram-negative bacteria, pathogenic RNA, pathogenic DNA, flagella, or peptidoglycan found in the cell wall of bacteria. There are about 11 different Toll-Like receptors. An example of Toll-Like receptors is TLR4. The ligand that binds to TLR4 is lipopolysaccharide, LPS, found on the cell wall of gram-negative bacteria. This is gram-negative bacteria. Here is LPS on the plasma membrane, which acts as a ligand for the Toll-Like receptor. When a ligand binds to the TLR, it undergoes a signal transduction. Protein kinases activate other proteins by adding phosphate groups. There is a cascade of protein kinases that stimulate transcription factors, which then promotes the expression of certain genes. For example, cytokines and interferons that fight viruses and promote inflammatory responses. Toll-Like receptors are involved in the responses to diseases such as asthma, sepsis, and atherosclerosis. Future prospects of Toll-Like receptors can be to produce vaccines for cancer, AIDS, or malaria by making TLR activators that stimulate dendritic cells, which will then produce a more effective immune response.
Whoa. Are Toll-Like receptors the only receptors involved in the immune system?
Well, Amanda, that's a good question.
Thanks.
Toll-Like receptors are classified in a family of receptors called pattern recognition receptors. These make up the innate immune response.
Where are they located?
Well, Amanda, these cells are strategically placed on the surface to recognize extracellular pathogens.
How do the receptors recognize what pathogens are bad?
Again, another good question.
Thank you.
The receptors use pattern recognition to detect PAMPs, which are also known as pathogen-associated molecular patterns, and DAMPs, also known as damage-associated molecular patterns. PAMPs are associated with microbial pathogens, and DAMPs are associated with the cell components that are released during cell damage or death. The receptors will bind to the structural shape, not with any micro precision, but they will be able to identify it as a foreign organism.
Wow.